same region, and also that you may remember Darwin was living. So a lot of happened over there. Why do I tell you this? Because this is very important in the whole picture of medical microbiology. We can discriminate three periods. The first period until the 1950s of classical techniques, culture, isolation of microorganisms. Then the second period in improving the same of these techniques. And then the third period, which is going on at this moment, is emergence of molecular techniques and molecular epidemiology. I will not discuss today on molecular epidemiology because I only have one lecture and it's far too much already, so I will skip that part. What do we think of when we think of microbiology? First, blood culture is a very important issue. Blood culture. Every patient coming into the hospital with fever will take a blood sample, and this blood sample will be cultured to see whether there are any bacteria present. Serological test and identification. So if you have any connection or any intervention with a specific microorganism, then we can see it in your immunological reactions. And of course, sensitivity to see which antibiotic treatment we should have. This is all until the 1980s. Then, in the 90s, there was automatic culture. So people didn't use anymore the plates on the lab, which is still used, by the way, but for blood culture not. And some very basic PCR techniques came in there. And from 2000, there's really a shift. And then I will go back to this period of Antonie van Leeuwenhoek, which took 100 years for development to Louis Pasteur, and if we look now, it took 100 years again to come to the major switch into molecular diagnostics. So that's more or less a kind of same period. And we are talking now about real-time PCR, Sanger sequencing, and DNA microarray technology. So what are these new developments? Blood culture techniques is completely automated now. Serologically, techniques are also automated. And then PCR, mostly homemade, so the laboratory itself made the whole test, especially for slow-growing microorganisms, because they benefit at the time, non-cultivable microorganisms, and yet unknown microorganisms. And also resistance is more and more detected on microarrays. And of course, some look in the near future with next generation sequencing and broad range PCR applications. So I will guide you through these 6 points during this lecture. Because the lecture is about molecular diagnostics, I won't go into details of the culturing techniques. But this is a machinery in which directly the blood of the patient comes in this kind of bottles, which are positioned in this automated system, and it just starts beeping when one of the bottles becomes positive. So that's what's currently used in the laboratory. This is quite an improvement because the liquid system, which is what I showed you, is far more rapid than a solid culture, which was used in the time of Louis Pasteur. So you get really an increase in time to result. So that means that you are now within 2 weeks for tuberculosis, you have a result. And that's rapid because before that it took up to 6 weeks before you had a result on tuberculosis. I will come to that later because we have now other techniques for that. Also, the serological tests, like the Legionella urine test, are based on the fact that antigens are presented in the urine of patients, and you can just put an ELISA on that, and then you will detect whether this patient has any connection to a Legionella infection. Of course, the problem with these techniques is that you don't know whether this patient had an actual infection or that is the remnants of an earlier infection. So that's the problem with serological tests. But what we are going to talk about today is molecular diagnostics, or its abbreviated as like this, MDX. And especially it's based, of course, the polymerase chain reaction. I won't explain the polymerase chain reaction to you because I think you are familiar with that. But there's some specific requirements for application in diagnostics. Of course, you all are familiar with positive controls, negative controls. But for diagnostics, you also need a process control, inhibition control. You need a confirmation of your targets, so 2 primers is not enough. You have to confirm that what is between those 2 primers is actually the sequence which you are looking for. So there should be a confirmation. And you must have a very specific target, so you get no intervention with other microorganisms, and a high sensitivity. Because when you have clinical samples, you want to detect even if there's only one bacterium present in it. And that's the basic which I come back to. When I look to the PCR, then you probably are familiar with real-time PCR or not? Yes. Well, this is this confirmation probe, and then you have the two primers, and then you get this specific reaction which gets fluorescence, and it's called the Tachman principle. The Tachman principle is based on the fact that the enzyme is cutting off this probe, and you probably know that in past days there was this Pac-Man. So this company called it the Tachman because it's the same principle of happing away all these nucleotides. So that you know that's where the name Tachman comes from. So this is this Tachman machine, which is completely automated, and you can do a closed system, keeping in this confirmation which is necessary for diagnostic purposes. This whole system is on the market since about 10 years. And it's really revolutionized the way clinical microbiological laboratories diagnose the human pathogens. Here you see the increase in publications in this field, and I can assure you, I didn't have a more recent picture, but it goes right straight up over there. Which means that there was really a need for this kind of application. So what do we do in the microbiological laboratory with this real-time technique? Of course, qualitative detection of microorganisms. So is the microorganism present in the clinical sample or not? But also quantitative detection, how much of this microorganism is present. Single nucleotide mutation for mutations leading to resistance to antibiotic treatment. Monitoring of infections and therapy. Are we given the right therapy, which means that the quantitative detection, if the therapy is right, will lower, and you can measure that in time. You can use all clinical samples, also for DNA or RNA, so you can use it for bacteria, viruses, parasites, fungi, so it doesn't matter anymore. You can even use formalin fixated biopsies from pathology departments. And you can detect non-cultivable microorganisms. So we find really new things now, and there are a lot of other items to go for. So it's not one test. I'll just give you an example of how these tests are functioning. For example, for detection of Mycobacterium tuberculosis, we have this real-time PCR, and you find on the DNA a very specific region which is only present in tuberculosis. You develop two primers and this probe, which is the real-time principle. Then you really know that when this is positive, that Mycobacterium tuberculosis is in your sample. But when it's negative, are you sure that there's no Mycobacterium tuberculosis in the sample? And that's very important because, especially in the Netherlands and also in Europe, most samples which you get for mycobacteria are negative, because the prevalence of positive mycobacteria is only about 2 to 3%. So if this is negative, this reaction, you really don't know whether this reaction from this patient in this sample has worked out well. So that's why we introduce an inhibition control. What we do for sensitivity is first, when we have this PCR, when you start using it, you want to know if it's really sensitive enough. So you have dilution samples, and you can just take out the concentration until one bacterium is still present, because above this, the crossing point above this line is a positive signal. So you can see that the dynamic range of this PCR is very large with the target I just showed you, from 1 bacterium until 10 to the 5th, approximately. Then you can put this in another calculation. If you know that this is 1, that is the crossing point, then you can make a standard curve of that. And if you have an unknown sample from a patient, you can just position that on the same level, and then you know exactly how much sample is in there. If you have two concentrations of real species, then you get this kind of analysis. So this is what I showed you before. But now, if you want to use this for clinical purposes, then you must know that there's really no intervention with any microorganism present in the throat or in the lungs or whatsoever. We don't know even all these species. So you have to use a huge number of clinical samples from patients, from healthy people, etc., to really show that they're all negative, whereas the only positives in this is the two which you added. So this is specificity. You really must be sure that there's no intervention with other microorganisms. And then we have all these controls. I won't go in detail on these controls, but I want to focus on this inhibition control, which I mentioned earlier. What you want to know is that when you have this target over here, that you're really sure that the test is going well. And when is the test going well? When the two PCR primers are really amplifying something. So what we did, we took this restriction enzyme site and we put in this new artificial sequence and put there a second probe on, which is only specific for this sequence. And this probe has another color than this probe. So we have two colors with the same PCR primers. And what's the reason for that? The reason is that you have now put this in another species of Mycobacterium, Mycobacterium smegmatis, which has this specific fragment is not present over there. But now it is, but it's in between another part. So what you get now an amplification of 132 base pairs from this clone and 67 base pairs from this. And this is red in the real time, and this is orange. So when the sample is positive, then you will get the orange signal. But when the sample is negative, then you should get a red signal. So in that case, you really are sure that when there was any Mycobacterium present in the clinical sample of this patient at that moment, then it is really negative, because this is positive and it really only amplifies when everything is okay in this reaction. But it also introduces another problem. And the problem is that you make use of the same PCR primers, which are also detecting the Mycobacterium. So there's competition between those targets. And you must be sure that this competition is okay. So when we add a certain fixed amount of this clone, then you will get signals like this. They're all the same, because you add exactly the same of this red target. And the crossing point is at cycle number 36. When you add your tuberculosis DNA to that, and you see here once again at cycle 36, but when you add a little bit of Mycobacteria, then you see that this signal is inhibited. And if you add more, then the signal is even further inhibited. So, and if this is your patient, and there's a lot of Mycobacterium present over there, this signal will become negative. But you know that this signal, which is one bacterium of Mycobacterium, is not influenced and is detectable in the presence of this clone. So now you are really sure that you have no problem with your sensitivity, you have your specificity okay, and now also you know really that there's no inhibition of the PCR reaction in this specific patient sample. Doing this, we found a lot of new infections. For example, Rhopalmia, Bartonella, and Trophorema, which are non-cultivable bacteria. And we always thought that they were only in specific cases in patients, but now we find that they are far more general. I will come to that later for Trophorema. For example, we had a 47-year-old male which has complaints of endocarditis, so his heart is not really optimal, and they found vegetations on the coronary artery valve. There were 16 blood cultures taken from this patient, and they were all negative. But the patient was given therapy, amoxicillin and gentamicin, but it failed. So it has to be surgery, and the coronary artery valve was replaced. This tissue was sent to the laboratory, and in histology and pathology department they showed infectious endocarditis. But then we introduced the PCR, and the PCR clearly identified Trophiringo which was previously not known to cause endocarditis. We never know, because it's not culturable. So this is the reason why these real-time PCRs are so in favor now, because you can detect far more. And if your controls are okay, then you really also know that what you're detecting is really something. Another technology which is coming to the department is the DNA microarray technology. We have very large synthetic oligonucleotides fixated on a glass surface, but these are very expensive. So in general microbiology, these large Affymetrix chips are not available. So they found a way to come across that. And they make a microarray at the bottom of the tube, very simple. And on this tube are different oligos spotted. And each oligo can detect specific bacteria. So for example, for fecal samples, you can detect which of these bacteria. But also, what you can do is you can put in here antibiotic resistances, and you probably are aware of MRSA, ESBL, etc. And also the probes for that are fixed on this bottom of this small tube. And this becomes very cheap, because now you can just have a very small detector over here, and here the spots are on the screen, and the combination of the spots really give you a result whether which bacteria is in it and which bacteria is resistant or not. So these are really the differences which can be made now based on microarray. Coming somewhat more to the viral case, a lot of Sanger sequencing is done. That's the classical way of sequencing. We do that for HIV genotyping, HCV genotyping, HBV genotyping, and also some drug-resistant strains we can find on mutations based in specific sequence over here. But they also can be performed by hybridization on a so-called strip, which is more or less an ELISA principle, in which you have hybridization and washing off, and if there's a good connection, then you will get a specific band. So these are really things which are used today in routine clinical microbiology. So all these sequences are screened, and you can really say this patient has a resistant strain or not, and then you can really decide which therapy should be followed. And this is based, once again, on viral load. So what you get then, if this is the time and this is the load of virus, then you can follow this HIV patient, and you see there's a very large increase over here, 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 and then they start therapy over here. So the triple therapy will start over here, and you see a little shift, and then it goes down. But at a certain moment over here, you can see a sudden rise, and that's the compliance of the patient. So the clinical doctor can say to the patient, well, we have a rise in your viral load, you really should comply to the therapy. And then you see the patient starts taking these pills again, and it decreases. And that's something which is monitored for every HIV or HCV positive patient, and also for other viruses like CMV or EBV. What's still not being done is to use this for bacteria, because we don't know whether this is a good approach for bacteria. To say, well, we have viral load, we can also talk about the bacterial load. And that's something which is being under discussion now. What we do now is that in bacterial infections, after phlebotomy, the decision has been taken to start therapy. But what you do afterwards, only a very minor part will really change the therapy. Because if the patient is going well, the doctor will not change the therapy. So we should have a very rapid test to really start the right therapy at the beginning. And can we do that with molecular techniques? Just skip this one. What we see here is the first example published in 2007. If you have the time over here, you have the temperature of the patient, and what you see here is the temperature of the patient. So it's about 37 degrees. So it's fever, and then there's blood culture, and the PCR is taken. Decreases, and it becomes fever again. Decreases, fever again. If you take the blood culture, it's positive positive over here, there, and there. If we take the DNA load, and this was specific as Staphylococcus aureus infection, and we see here that there's about 150 bacteria per ml in this patient. But you also see that the temperature raises and the DNA load also raises. So here you have this higher amount, and then it decreases when the temperature of the patient decreases. But over here you see that the temperature rises again, and also the DNA load of aureus rises again. However, in the clinics they decided to start flucloxacillin, which is specific for aureus, but because it did not function, they started ceftriaxone, which is not for aureus, it's for gram-negative bacteria. So they did a wrong decision over here, and the patient died at this moment. So if we had followed at that moment, which we didn't know of course, the real PCR, then probably the amount of Fluclox would be increased rather than adding ceftriaxone. So this is an example that really shows that we can do something with these loads. The same is when you look at the C-reactive protein, which is a marker for an infection in patients. You also see that the C-reactive protein increases very largely over here, but it goes almost in parallel with the DNA load. So that was another proof that really something is applicable with this bacterial DNA load. Last example, here you have 3 patients in which you really see now in time that the DNA load is really decreasing, so this is a very successful therapy, and after 4 days of treatment this was very successful, and all the patients recovered. Except for one. What we already discovered over here is that the therapy did not do its work quite properly. And then it decreases again, and then it increases, and here the patient died. So you get really a different picture when the therapy is not really going well. So this means that these new techniques are now validated if we can use them in routine clinical laboratories. A very new item is second generation sequencing, and I don't know whether you're familiar with that. Are you familiar with second generation sequencing? Looks so. But the second generation sequencing is quite different from the classical sequencing in a way that a huge number of sequences are generated, mostly short pieces, and the computer has to put them all together and make an alignment of them. So you may have from one specific sequence about 10,000 to 1,000,000 the same sequences. So you have to put them all together, and that for a long row of sequences. And there are numerous developments in that. And there are 3 major systems, like I mentioned here, the 454 pyro sequencing, the solid sequencing, and HiSeq MiSeq from Illumina. That are very important issues at this moment. And this principle is quite different. For example, you have so-called bridge PCR, and you have very small pieces on a glass plate, millions and millions on a quarter of a centimeter, and then your DNA fragment adheres to it and only can sequence 50 base pairs. But on the other side, this side, it also can sequence 50 base pairs. So you have a sequence from 50 and 50, but you know that they belong together because they're on one strand. So you have all kinds of different ends, and the hay is putting that together, millions and millions together. And then you get a very validated sequence of, let's say, the whole human genome, but also the whole bacterial genome. I won't go to the detail. Very important with this kind of techniques are 3 different ways looking at that. You have sequence depth, coverage depth, and read length. Sequence depth means the total number of all the sequences or reads, the sequence reads, base pairs, represented in a single sequencing experiment. So the more reliable the sequence is. Also the coverage depth, the total number of nucleotides from reads that are mapped to a given position. So you can have the same sequence 10 times, 20 times, 100 times, 1,000 times, and the more you have, of course, the better it is, the more reliable your sequence is. And then the read length, which is very short, between 50 at smallest until max 500 at the fire sequencing. But the fire sequencing is already going out of the system because it's not powerful enough. So the focus is on the short reads. And although these second generation sequences are not yet available in clinical applications, a third generation is coming out. And there are 3 companies working on that. And what's the difference now is that it's going further. Now we can sequence a single cell completely. So if you have only one cell or one bacteria, you can sequence the whole genome. And not only that, if you can do that from one cell, you can also see the difference within that cell, and you can say how much of these cells are present in a total environment. So how many bacteria are present. So it will become quantitative. So the most ideal picture. But there are also huge problems with it, because it's hard to generate clean data. Files with quality scores. That's very, very difficult to get. Dealing with sequencing errors. Huge. Interpretation of data, we need to correctly align the sequence, it's very difficult. And a lot of other issues. I will skip this. Oh, but we can now sequence a $1,000 genome, so that's really get a decrease in this sequence. So you get an impression how fast this is going in the past 10 years. And we know that Greg Venter was the first person whose genome was sequenced. Cost about $70 million, and James Watson was about $1 million, whereas now we can do it for $1,000. So get an impression. The last thing I want to discuss with you, because we run out of time, is the so-called new applications broad range PCR. In that case, you don't bother what bacteria is in it, you just do a normal PCR on an interspatial region, and then you really find what's in there. And how does it function? It's very simple. You have the bacterial genome, and these interspatial regions may be present 4 times in the genome, but they are different in length, but constant different in length. So for Enterococcus faecalis, you always end up with 2 fragments, 377 base pairs and 275 base pairs. So that means that this is our pattern of faecalis. So although you didn't know that faecalis was in there before, if you find this, then the computer says to you, this is Enterococcus faecalis. So that's the principle behind it. These are the PCRs performed, and this can be performed in every laboratory. There are now 3 different PCRs with 3 colors, and they are just normally amplified like you do with normal PCR. This is, I think for the time, it is a bit difficult to say. And here you have a specific example of all kinds of peaks from the gut flora, and there's one tiny peak over here, which is Trophima whipplei. So this was a patient who was complaining for 10 years and had lost about 25 kilograms of weight, and they had no diagnosis, and based on this small peak over here, it was diagnosed for Trophima whipplei. So what we also did is look in students whether the gut flora is constant in time. And what we saw over here is that in time we had some differences. You can see here all these colors correspond to, and all these peaks correspond to specific bacterial species. But you can see that although this is the same patient or the same student, this is quite different from this, and this is quite different from that. And so we did find some difference. But in the end, the same pattern came back. So this is white pattern in red, which resembles this pattern, whereas the other patterns resemble more to each other. But when we put the time frame on that, then there was an explanation, because every time he started to drink, the gut flora changes. But we know now that if you start to drink alcohol, it takes about 3 days before your gut flora is back to normal. And it's stable. Person-wise, it's stable. And we can discriminate then a lot of species in your gut flora from this one single PCR. Then you make a heat map of it. And what you see over here is that we made a heat map of patients ill and patients healthy. But if you look at all these different species which are present in the gut flora, then you can You can really not make sense of it. But that's because you are looking too deep into the species level. So you can also, in this technique, go a little higher at the family level, or even at the phylum level, which is the highest level of bacteria. And if you look then at the same picture, then you really see that there's a good discrimination between diseased and healthy. But what you also see, that this reason for diseased and healthy, if you look at all the major groups which are now represented over here, that is in this group, the firmicutes. In these firmicutes you really see difference between the diseased and healthy. So there's something going on in this group which you can recognize now. And then you can zoom back specific on this group and see what's happening over there. So this is more or less the overview that you can go from this high level to this very species level and get more and more complex support. For the doctors, this is the picture. What we developed is that you can now follow in time a specific patient, and you can put over here the antibiotic treatment, and you really can see now what the influence is of antibiotic treatment on specific species, because every line over here is a specific species, and you can follow it in time. So we are now able, with these new molecular diagnostic techniques, to really show what we are doing. We treat every patient, but we really don't know what the antibiotics are doing, and if it's the right antibiotics. Molecular diagnostics makes it possible now to really introduce these measurements on it. And we hope that we will end up with saying what's healthy and what's diseased, and if it's diseased, that we really know what we should do to go back from a disease state to a healthy state. And that may be by using antibiotics, it may also by using probiotics or food or whatever, other supplies, but we really can show now what's happening. So and that's a major difference in medical microbiology nowadays with these techniques. So in, let's say, 200 years, we have the first 100 years with culture, and now the expectation is that the next 100 years will be with molecular techniques. So I want to end up with this statement that readily cultivated bacteria draw importance to themselves, whereas the other forms, being less docile or even resistant, escape as tension. But now we're going to catch them with the new molecular diagnostics which we are developing now. And I realized it was a huge overview, too much for one lecture in fact, but it's a start, let's say it's a start. So I hope you really did get something of it, and we will fine-tune this, and please respond to us that we can optimize this lecture, so that you are the first victims following this. Thank you for your attention. Are there any questions?